In the kidney, the majority of filtered glucose is reabsorbed by the sodium glucose transporter SGLT2 in the proximal tubule. Residual glucose that passes the proximal tubule into the distal tubule is reabsorbed by the sodium glucose transporter 1 or SGLT1 transporter. The majority, however, is reabsorbed by SGLT2. It was this observation and observations in human genetics in subjects that had mutations in the SGLT2 genes that showed these subjects had renal glucosuria that showed that SGLT2 was a potential target for type 2 diabetes. Simply, the idea was if you could inhibit SGLT2, you would inhibit the reabsorption of the glucose in the kidney, and you would simply excrete glucose into the urine.
Remogliflozin etabonate, which we'll refer to as REMO, was originally discovered in the laboratories of our Japanese collaborator, Kissei Pharmaceuticals. Kissei initially did some early non-clinical and clinical work which demonstrated the potential of REMO as an anti-diabetic treatment. Kissei subsequently licensed REMO to GlaxoSmithKline, where I was on the project leadership team, until Glaxo made the unpopular decision to give the program back. This decision was made due to internal factors within the company and had nothing to do with the safety and efficacy of this asset. At that point, I realized that REMO could be a real drug for the treatment of type 2 diabetes. So I left GSK and started Brighthaven Ventures, a subsidiary of Avalent, to further the development of REMO.
For the initial discovery of remogliflozin, Kissei Pharmaceuticals used an entirely in vitro screening assay where they used a number of small molecules to look for inhibitors of SGLT2. At the same time, these inhibitors were not to inhibit SGLT1, so they were looking for highly specific and highly selective SGLT2 inhibitors. During this process, they were able to identify a number of compounds that would inhibit SGLT2. And after further development and a number of different selection criteria, Kissei decided to further develop remogliflozin for preclinical development. But right prior to the preclinical development, Kissei did some in vitro assays in cell-based systems where they took cells and transfected them with either SGLT2 or SGLT1 and treated the cells with the inhibitors and looked for selectivity and specificity of the inhibitors. And what they found was that remogliflozin showed the best profile for these characteristics. Indeed, remogliflozin has a very high selectivity and a very high potency for SGLT2 over SGLT1. All of the required FDA safety and toxicity studies for REMO have been completed. This includes studies for carcinogenicity and toxicity in long-term studies, even up to 2 years. And in all the studies that have been conducted, REMO is found to be very safe and very well tolerated. As the development of REMO progressed, it moved into non-clinical or animal model studies. In a number of different studies, both in healthy animals and in animals with type 2 diabetes, REMO was orally administered and shown to be very safe and well tolerated in animals up to very high concentrations of drug. It was shown that REMO could induce glucosuria, renal glucose excretion, as well as reduce fasting plasma glucose and A1C in type 2 diabetic models. Prior to Glenmark's recent successful phase 3 clinical study, REMO was examined in a number of other clinical studies. There were 17 phase 1 studies, 2 phase 2A studies, and 2 phase 2B studies. In the phase 1 studies, there was primary focus on pharmacokinetics and pharmacodynamics. REMO was shown to be very safe and well tolerated in those studies, even at doses up to 4,000 milligrams a day. The phase 2A studies were conducted in primarily type 2 diabetic patients. In these studies, Remo was examined for initial effects on HbA1c and fasting plasma glucose. It met all of its primary endpoints in these studies. In the phase 2b studies, which were conducted with a larger population of type 2 diabetic patients, these studies primarily focused on dosing with Remo. Again, the primary endpoints were met, which was lowering of HbA1c. And in addition to that, there was a number of secondary endpoints that were met: reduced fasting plasma glucose, reduction in body weight, reduction in blood pressure, as well as an increase in insulin sensitivity. Some other criteria that we looked at in retrospectively, we found that in the phase 2b studies, as well as the BRID study, which I'll speak to in just a moment, that these subjects showed, that were treated with Remo, showed reductions in liver enzymes. And in fact, when we did a further analysis of this, we showed that these subjects had reductions in NAFLD fibrosis scores as well as FIB4 scores, indicative that Remo actually would have a positive effect in subjects with fatty liver disease or NASH. More recently, we developed a biphasic formulation or an extended release formulation of remogliflozin in order to provide a once daily dosing of the drug. And in a third phase 2b study, which was entitled the BRID study, we examined this biphasic formulation in type 2 diabetic patients. And similar to the other phase 2b studies, in this case the biphasic formulation was very comparable in showing reduction in HbA1c and hitting all the other secondary endpoints, such as a reduction in fasting plasma glucose and reduction in blood pressure.
So how does Remo compare to other SGLT2 inhibitors that are also on the market? Remo works to the same mechanism of action as all these other SGLT2 inhibitors. Appears to have very similar abilities to lower HbA1c, fasting plasma glucose, causes weight loss, causes a reduction in blood pressure. So in many ways, Remo is very similar to these other drugs that are on the market. But Remo does differ in a couple of ways very distinctly from these other compounds. First of all, Remo's chemistry is very different than the other SGLT2 inhibitors. Remo has an inherent antioxidant activity that's based on the chemistry that that creates Remo. So this inherent antioxidant activity, we think, also plays a role in the potential for the treatment of other diseases such as NASH and fatty liver disease. Remo also shows a very strong ability to lower blood pressure, maybe more so than the other SGLT2 inhibitors. So we are looking to bring Remo as an anti-diabetic to several other countries. However, in the United States, Amylin is focused on bringing Remo forward for the treatment of NASH and fatty liver disease as a monotherapy and also potentially as a combination therapy or fixed-dose combination with other compounds. Remo has already demonstrated a number of attributes that suggest that it would be very effective in the treatment of NASH. It has a very strong anti-inflammatory activity in the liver. It also, as we've discussed already, talked about the antioxidant activity that Remo has inherently as a molecule. Remo also causes weight loss, which is an important component of treating NASH. And then finally, Remo is an excellent insulin sensitizer, and reducing insulin resistance, particularly in the liver, is a critical part of the overall therapy in terms of trying to reduce NASH. Remogliflozin is a safe, well-tolerated, and effective treatment for type 2 diabetes. I've been with this project from the very beginning, and I'm extremely excited to be able to bring this drug to patients with Glenmark in a way that truly changes their lives for the better.